Good morning, and welcome to Mersana Therapeutics' second quarter 2025 financial results and business updates conference call and webcast. Currently, all participants are in listen-only mode. There will be a question-and-answer session at the end of this call. Please note this call is being recorded. I would now like to turn the call over to Jason Fredette, Senior Vice President, Investor Relations and Corporate Communications.
Thank you, Operator, and good morning, everyone. Before we begin, please note that this call will contain forward-looking statements within the meaning of federal securities laws. These statements may include, but are not limited to, those related to the potential clinical benefits of our product candidates and platforms, our clinical trial progress and design, addressable market opportunities, and anticipated clinical milestones and data presentations, and cash runway. Each of these forward-looking statements is subject to risks and uncertainties that could cause actual results to differ materially from those projected in such statements. These risks and uncertainties are discussed in our quarterly report on Form 10-Q filed with the Securities and Exchange Commission on May 15, 2025, and in subsequent SEC filings. Our filings are available at sec.gov and on our website, mersana.com. Except as required by law, we assume no obligation to update forward-looking statements publicly, even if new information becomes available in the future. On today's call, we have Mersana's Chief Executive Officer, Dr. Marty Huber, and our Chief Operating Officer and Chief Financial Officer, Brian DeSchuytner. With that, let me turn the call over to Marty to begin the discussion.
Thank you, Jason, and good morning, everyone. The second quarter of 2025 was eventful for Mersana, particularly as it relates to Emi-Le, our Dolasynthen ADC targeting B7-H4. Chief among the highlights were our oral presentations at ASCO 2025 and ESMO Breast Cancer 2025, and the strong enrollment progress we made in our expansion cohorts. At ASCO in Chicago, Dr. Erica Hamilton of the Sarah Cannon Research Institute presented data as of March 8 data cutoff across all patients who are enrolled in our Emi-Le phase 1 dose escalation and backfill cohorts. Safety and tolerability remained consistent with previously reported data. This presentation also included clinical activity data across all enrolled B7-H4 expressing tumors. At intermediate doses ranging from about 38 milligrams per meter squared to 67 milligrams per meter squared, the confirmed objective response rate, or ORR, was 31% among evaluable patients with B7-H4 high tumor expression, which we initially defined as a tumor proportion score of 70% or higher. The presentation also highlighted some intriguing activity that we have seen in adenoid cystic carcinoma type 1, or ACC1. This is a rare head and neck cancer with a very poor prognosis and no approved therapies. In the past, other development candidates, such as VEGF TKIs and Notch inhibitors, have investigated this tumor type and have shown response rates ranging from the mid-single digits to the mid-teens. Among the 9 evaluable patients with ACC1 who received any dose of Emi-Le regardless of B7-H4 expression, we observed 4 confirmed responses and 1 unconfirmed objective response. And subsequent to the March 8th cutoff, that 1 UPR was confirmed for an objective response rate in ACC1 of 56%. In recent months, we have enrolled additional patients with ACC1 in backfill cohorts. While these data help to demonstrate Emi-Le's broader development potential, our focus today is on addressing the significant unmet needs of patients with triple-negative breast cancer, or TNBC, who have previously been treated with a topoisomerase I inhibitor, or topo-1 ADC, such as Trodelvy, Enhertu, or Datroway. The standard of care today for these patients is single-agent chemotherapy, and unfortunately, their expected outcomes are exceedingly poor. Based on reported data from the original ASCENT phase 3 trial of Trodelvy in topo-naive patients with recurrent metastatic TNBC, the objective response rate, or ORR, for single-agent chemotherapy was 5%. Progression-free survival, or PFS, was 1.7 months, and overall survival, or OS, was 6.7 months. At the ESMO Breast Cancer Congress in Munich, Germany, Dr. Hamilton presented data as of that same March 8th cutoff from patients with TNBC who had received intermediate doses of Emi-Le, highlighting patients who had received 1 to 4 prior lines of treatment. Nearly all of these patients also had received at least one previous topo-1 ADC. Among the patients with B7-H4 low TNBC who received an intermediate dose of Emi-Le, observed clinical activity resembled today's standard of care with an ORR of 0, a median PFS of 6.4 weeks, and a median OS of 5.7 months. But among those patients we have initially characterized as B7-H4 high TNBC who received an intermediate dose of Emi-Le, the ORR was 29%, the median PFS was 16 weeks, and the median OS had not yet been reached as of the data cutoff. It's data like these that led us to initiate dose expansion, and in recent months we have continued to make progress in this phase of development. In expansion, we are enrolling patients with TNBC who have received 1 to 4 prior lines of treatment in the metastatic setting, including at least 1 prior topo-1 ADC. We're investigating 2 Emi-Le dosing regimens. Our dose A cohort is receiving a 67.4 milligram per meter squared dose of Emi-Le every 4 weeks. And our dose B cohort is receiving 80 milligrams per meter squared dose of Emi-Le every 4 weeks, following a loading dose of 44.5 milligrams per meter squared on days 1 and 8 of the first 4-week cycle. Collectively, we have enrolled more than 45 patients across these 2 cohorts, and we remain on track to report initial clinical data from expansion in the second half of 2025. Now, finally, on the Emi-Le front, we are often asked how big the post-topo-1 TNBC opportunity is. We believe it is sizable, and it has the potential to get substantially larger. As a reminder, today, topo-1 ADCs with TNBC indications are only approved in the recurrent setting. Despite this, Trodelvy is already expected to generate about $1 billion in global TNBC revenues in 2025. Of course, this figure does not include revenues that other topo-1 ADCs are generating in this setting, nor does it consider how the opportunity would increase as these agents move into early lines of therapy. For example, we believe the recent positive readouts of ASCENT-3 and ASCENT-4 will make Trodelvy the new frontline standard of care for patients with TNBC, which will greatly increase the post-topo-1 population of patients. As a reminder, emerging clinical data suggests that once a patient receives an initial topo-1 ADC, a subsequent topo-1 agent has substantially reduced benefit due to payload resistance. We believe this evolving treatment landscape opens the opportunity for a novel non-topo-1 agent like Emi-Le to address the growing unmet need for topo-experienced patients if it is approved. So if a patient receives Trodelvy in the front line, Emi-Le could potentially be used as a second-line therapy. Or if a patient receives another topo-1 ADC, such as Enhertu, Dato-DXd, or Sac-TMT in the recurrent setting, Emi-Le could potentially be used subsequent to that. Additionally, because of Emi-Le's differentiated tolerability profile, we believe it could potentially be explored in combinations with other agents, including topo-1 ADCs, platinum chemotherapy, and PD-L1 agents. So let's move on to XMT-2056, Mersana's Immunosynthen ADC targeting HER2. As a reminder, GSK has an exclusive global license option to co-develop and commercialize this candidate. XMT-2056 is in the dose escalation portion of our phase 1 clinical trial, which is enrolling patients with a variety of HER2-expressing tumors. And we are pleased to report that in July, we achieved a $15 million development milestone under our agreement with GSK. Payment of this development milestone is due later this quarter. With that, let's turn the call over to Brian for our financial review.
Thank you, Marty. Beginning with our balance sheet, we ended the second quarter of 2025 with $77 million in cash and cash equivalents. Net cash used in operating activities for the second quarter of 2025 was $22.6 million, which included $2.4 million in severance-related payments. And in July, we paid approximately $17.9 million to pay off our debt facility. We continue to expect that our capital resources will enable us to support our current operating plan commitments into mid-2026. Please note that our cash runway guidance does not assume the receipt of any future milestone payments from our current collaborations or proceeds that we may realize from future collaborations. Collaboration revenue for the second quarter of 2025 was $3.1 million compared to $2.3 million for the same period in 2024. The year-over-year change was primarily related to increased revenue recognized under our collaboration and license agreements with J&J and Merck KGaA, partially offset by reduced revenue recognized under our agreement with GSK. Research and development expenses for the second quarter of 2025 were $16.2 million, compared to $17.2 million for the same period in 2024. For the most recent quarter, approximately $900,000 of this spending was related to non-cash stock-based compensation. The year-over-year change in R&D expense was primarily related to lower headcount and related employee compensation costs, which were partially offset by an increase in costs related to Emily and XM2056 clinical development activities and manufacturing activities associated with the company's collaborations. General and administrative expenses for the second quarter of 2025 declined to $7.4 million compared to $10.5 million during the same period in 2024. Approximately $1.1 million in non-cash stock-based compensation expenses were included in G&A for the most recent quarter. The year-over-year change in G&A was primarily related to lower headcount and related employee compensation costs and a reduction in consulting and professional services fees. During the second quarter, we incurred $3.9 million in restructuring charges related primarily to severance and benefit payments, outplacement services, and related expenses. And finally, Mersana's net loss for the second quarter of both 2025 and 2024 was $24.3 million. That concludes our business update. Operator, would you please open the call to questions from the audience?
We will now begin the question and answer session. To ask a question, you may press star, then one on your touch-tone phone. To withdraw your question, please press star, then two. At this time, we will pause momentarily to assemble our roster. And the first question comes from Tara Bancroft with TD Cowen. Please go ahead.
Hi, good morning, everyone. So my question is, for the data that are coming in imminently in the second half, can you help us set expectations for what will actually be in the data, like maybe minimum amount of follow-up that we should expect from patients that are enrolled, and if all of the 45 that are enrolled are going to be included in that data set and or available, and what benchmarks that you would want to meet or beat? Thanks so much.
Thank you, Tara. Good morning. There was a lot there. Let me try to walk through each of those. Once again, we've now enrolled over 45 patients. We would expect that group of patients to be the basis of the data, but we will have to be a little careful about committing to an exact number because we have to, treat the patients, get through it, get scans, etc. With regards to dose A and B, the one thing I would remind you is it's going to include both dose A and dose B. But just to remind you, dose A started at the beginning of the year. Dose B, as we announced in May that we had started enrolling patients at dose B. So the one point I would make is that the duration of follow-up will be longer on dose A than dose B, but you should have sufficient data on dose B to understand, efficacy, safety, durability, etc. And so you will be able to, so kind of your standard presentation like we've done before. And then the final part of that is just to remind you, these are all patients, and we would generally expect that about 40 to 50% of them will be in the B7H4 high bucket.
Okay, got it. And just, I know you don't typically set expectations for efficacy, but maybe just a good benchmark for what you think would be a good result.
Well, I think just to remind you that the efficacy for the standard of care in this setting is a 5% response rate and a 6 to 7 week PFS. So like the data we shared at Munich, we would think that's kind of the minimum bar, that if you have the 16 weeks and you have a response rate in the 20s, we believe that you would beat the standard of care in a randomized trial fairly readily. Better than that is obviously better, but I think that's kind of the minimum bar that we feel confident moving forward with.
Okay, great. Yeah, thank you for that. That's very helpful.
And your next question comes from Jonathan Chang with Leerink. Please go ahead.
Hey, this is Ewan on for Jonathan. Thanks for taking our questions, and congrats on the enrollment progress. Can you just provide more color on the implementation of the proteinuria mitigation strategies in the dose expansion cohorts? Thank you.
Sure. So just to remind people, is Amendment 5 was put in place at the beginning of this year. What we did in Amendment 5 was, one, we implemented a prospective, in other words, routinely starting in patients, either an ACE inhibitor or an ARB, before they even, at the beginning of treatment. So the idea was to try to minimize the development of proteinuria. The major change, however, was since we had observed that the proteinuria was primarily albuminuria, isolated albuminuria, in the absence of changes in serum creatinine or serum albumin, with the consultation with our onconephrologist, the protocol amendment is allowed to continue to treat patients as opposed to interrupting treatment, because previously it had this kind of protocol mandated interruption. So what we will be able to present you data on is with the updated data set for dose B, is at the higher dose, have we been effective in preventing those treatment delays?
Very helpful. Thank you.
Again, if you have a question, please press star, then 1. The next question comes from Charles Zhu with LifeSci Capital. Please go ahead.
Hey, good morning, everyone. Thanks for the call and congratulations on the updates and the enrollment across the board. Couple from me. Want to confirm if you've settled in on this TPS of 70 as a B7H4 high versus a low cutoff. And maybe as a follow-up to the prior question, with respect to the proteinuria mitigation, how would you evaluate, like between those two doses, if we're, talking about something like, what kind of a delta in clinical profile, I guess, would justify the additional step of undergoing mitigation steps, go for the more efficacious as opposed to a less efficacious dose? Thank you.
With regards to your first question, the expansion data set will be part of the data set that we use in finalizing the cut point. I think suffice it to say, even if the TPS score moves a little bit, we fully anticipate, based on the literature and our initial backfill and escalation data, that we're talking 40 to 50% of the patient population, even if the actual TPS score to define that population changes a little. So we haven't locked in on TPS 70 today, but that'll be something that we'll use this data set to do. Excuse me. With regards to proteinuria, ultimately, our hypothesis that came forward out of our initial data set was that these higher doses were resulting in more patients having more than a 30% reduction or getting that initial UPR. So our core hypothesis is that if you don't have the treatment interruptions and more patients are getting a reduction in the tumor, ultimately that will lead to a better outcome for patients, more responses or better PFS, et cetera. So what we will be looking for in B is that we are able to maintain patients on drug, and that will give us better efficacy. If that efficacy trade-off is not good enough, then dose A would be the go-forward dose. But that's a data-driven decision that we'll take. In fact, these data are a critical part of that decision process. So we need to get the dose A and dose B data fully in hand.
Understood. Thank you for taking the questions and congrats again.
And your next question comes from Michael Schmidt with Guggenheim. Please go ahead.
Hey, good morning. Thanks for taking my questions. Just a couple of follow-ups on the expansion cohorts. Did you say roughly how many patients would be in dose cohort A versus B for this upcoming update in the second half? And what type of venue are you planning to do this at? And then I had a follow-up.
I'll answer the venue question first. With regards to the venue, we have not stated whether this will be a corporate company presentation or a scientific meeting. That's a decision that will be TBD. With regards to how many patients are in each, we haven't provided that degree of specificity, but we'll note that we've been enrolling the dose B patients now for several months. So I think suffice to say there should be a reasonable number of patients at both doses. The only major difference will be the duration of follow-up will be shorter for the dose B patients than dose A patients.
Makes sense. Thanks. And then a question just on possible next steps as we think about potential phase 3 study in TNBC. So just in terms of trial design, should we think about a study that may look similar to the ASCENT study perhaps? And then would you limit enrollment to topo-exposed patients only or also allow naive patients? And if so, I guess why not? Or why wouldn't you include naive patients as well? Just your estimation in terms of what percentage of patients do you think are topo exposed in the third line and later setting at this point? Thanks.
Well, I think I'll take the last one first on the topo exposed. To be frank, whether we allow the topo naive or not is sort of a moot point in second or third line TNBC in the United States today, because as you recall from our demographics, almost all of our patients had seen a prior topo. So I think that makes it, once again, the detail of do you allow them becomes kind of irrelevant because from a standard of care point of view, they're all going to have received them, or almost all. With regard, I think as we see the ASCENT 3 and 4 data come out, that's going to even increase because now they're going to get them first line. So I think the big change we'll see is there's going to be second line patients that historically would be just getting a topo. The second line patients now will be post topo. So, our ideal, one of the reasons why do you say post topo is a couple things. One, we have a fast track designation in post topo breast cancer. And the way our fast track designation reads, it actually allows you to get a little broader than classic TNBC, because remember we have the HER2 lows. So if they've seen like in HER2, that counts. Now, obviously all of this is a discussion with the agency, but we think we have this unique opportunity in this post topo setting based on the unique properties of our platform. So this is kind of an easy space just to go into, given that most patients are going to be in this space anyway.
And then, maybe just to follow up, I guess, is there an opportunity to perhaps do a study that includes patients beyond the classic TNBC definition, including, for example, HER2 low patients into the same study to expand the opportunity?
Well, I think, and that's one of the advantages of doing a randomized trial because you'll have a control arm. We have no agency, conversations yet on this, but if you think about it, one of the limitations, for example, if you do a single arm, you have to make sure then that every patient that enrolls has seen every potential prior therapy. Whereas if you go into a randomized trial, you can be a little looser on that because the control arm will account for that. So we do think, doing a randomized trial, you can do it pretty much just as fast because, your PFS endpoint, your control arm is in 6 weeks. You also get your survival data. You avoid doing a confirmatory trial. And oh, by the way, you might be able then to broaden out your population exactly as you're suggesting.
Great. Thank you.
And your next question comes from Colleen Kusy with Baird. Please go ahead.
Hey, good morning. This is Nick on Colleen. Thanks for taking our questions. Just wanted to ask for Emily, can you comment on how the rate of enrollment is going versus your expectations and whether you plan on opening any additional sites for the expansion? And I had a follow-up after that. Thanks.
Right now it's going very well. I think for a TNBC population at a limited number of sites, getting 45 patients who are, these are patients who we're having to get biomarker data on. So these are all patients who have to get a tissue result tested, et cetera. Had to have had a prior topo
be 1 to 4 prior lines. So it is, I think we're very pleased with the outcome of the enrollment. We don't envision needing additional sites to finish expansion. Certainly as we move into a pivotal, though, we're going to want to, expand to a broader population.
Great, thank you. And then moving to 2056, I know you commented earlier that enrollment had been a little slower just to some competition in the area. Could you comment on how the enrollment's going there as well, and if you have a decision on which indication to go in with that readout?
Well, just, we haven't selected, obviously, an indication for 2056, other than it's HER2 positive cancers. But I think one of the reasons we are very, very pleased to discuss the 15 million milestone is this is a program that's been very, very quiet, and we hadn't had a lot of progress. In fact, we were kind of, in our last call, kind of downplaying the enrollment a little bit. It picked up, and we made our milestone. So, in our view, it's chugging along, and I think we're still maintaining our guidance that we'd like. We're not committing to sharing dose escalation data this year, but we are hoping to share some data, pharmacodynamic data, showing that the mechanism of action is in fact working in patients.
Great, thank you so much.
Again, if you have a question, please press star, then 1. Your next question comes from Andy Hsieh with William Blair. Please go ahead.
Thanks for taking our questions. So I think you have a couple of months of experience in terms of the trial protocol change to address proteinuria. And I'm curious if you have heard from investigators any feedback regarding the change and potential improvement to allow patients to stay on therapy. So that's question number 1. Question number 2, just kind of housekeeping, maybe a follow-up to the previous question. So the 15 million milestone payment, is that completely progress-based, or we can glean into potential, promising efficacy there as well? Thank you.
So I'm gonna answer the question on Emily, then I'm gonna turn over to Brian to talk about the criteria for the 15 million. So with regards to the proteinuria, I think all we can say is that enrollment's continuing at dose B. The investigator enthusiasm remains high. And then we're looking forward to sharing the data.
Yeah, and with respect to the milestone, it's a development milestone, but unfortunately we can't get into additional detail on that. It remains confidential.
That's helpful. Thank you.
And your next question comes from Asthika Goonewardene with Truist. Please go ahead.
Hey, good morning, guys, and thanks for taking my questions. So, Marty, I think you hit the nail on the head with the whole idea of dose B and trying to see if you're able to keep the patients on therapy longer, which I think is exactly what you need to be doing with an ADC here. But let me ask you this: when you present the data or announce the data later on this year, do you think you'll have that question answered? I.e., does, with the prophylaxis and everything going into play, that you are able to keep the patient on therapy longer on dose level B? Will that be answered this year? And then I got a couple of follow-ups.
Yeah. And while dose B will have more limited durability, I think, just to remind you, we were seeing the treatment interruptions in cycle 2 and cycle 3. So we're confident that we'll have sufficient data to at least answer that, show the difference between what was happening at the higher doses pre-Amendment 5 versus with the new proteinuria mitigation strategy. So I think we're pretty confident in our second half data is like we should be able to answer that question.
Got it. Excellent. And then just technicality here, what proportion of the TNBC patients that you recruited have actually seen Enhertu, and how do you see that play out as you're thinking about recruiting a pivotal study? Obviously, we're going to get Trodelvy in that frontline setting kind of impacting here, but do you think you'll see patients who've seen both Trodelvy as well as Enhertu?
Well, just to remind you, in our initial demographics that we presented previously, a quarter of patients had both. I mean, think about that. 27, I think it was 27% of patients had seen both Trodelvy and Enhertu. Now, is that going to be the exact same in our study going forward? It may be a little lower because what we suspect, given the limited outcomes with that strategy, if we have a pivotal study open that allows patients to go on whether they've had either Trodelvy or Enhertu, we think a fair amount of those patients who come in with the HER2 low will then have an opportunity to get the Enhertu and then go on to our trial. Now, once again, this is something we'll be part of the details of study design when we engage with regulatory authorities. But that is certainly something we are entertaining.
Got it. Thanks. And then we talked a little bit about some other breast cancer indications, but so let me ask you just point blank, Marty. The next steps and going into a pivotal study, will you be kind of maybe focusing exclusively on TNBC, or do you want to do some more work to kind of get some of these other areas of breast cancer, maybe the HER2 lows, generate some more data on that, and kind of also kick off a pivotal study that encompasses that patient population as well? Thanks, guys.
We would love to do additional activities, including hormone receptor positive breast cancer. I think there are opportunities to study that. At this point in time, given our current kind of data and capital constraints, we are focused on TNBC. But what I want to remind you is, in our fast track designation, it's not limited to TNBC. It does include these HER2 lows. So I think we have to think about it. This is where we probably need to just think about the populational difference of a classic TNBC. Well, in a way, we're thinking of as post-topo breast cancer is actually kind of a wider population than just TNBC. So this will be a critical part of our discussion of the details of design. And we do have data in these HER2 low post-Enhertu patients. So this will be something to look forward to.
This concludes our question and answer session. I would like to turn the conference back over to Dr. Marty Huber for any closing remarks.
Thank you, operator, and thanks everyone for dialing in. We hope you enjoy the end of the summer. That concludes our call, operator.
The conference is now concluded. Thank you for attending today's presentation. You may now disconnect.